Hello everyone, good morning. So, we were doing the dicotyledonous root in which we have seen this structure. Lateral roots generally arise from a pericycle, through therefore they are endogenous in origin, that is, they are developed from inside. Vascular bundle is radial, that I have already explained in the previous video. Xylem and phloem bundle are equal in number, and basically the conjunctive tissue, that is the parenchymatous tissue, lies between xylem and phloem bundle. So all the tissues on the inner side of endodermis such as pericycle, vascular bundle and pith constitute stele. These three structures, pericycle, vascular bundle and pith, they together form a stele, and the central part of a stele is made up of parenchyma cells called as pith. That is called as pith. Now we will do monocotyledonous root. You can see this is the diagram of TS of monocot root, in which we can see the clearly epidermis, and from epidermis the appendages are present which are called as root hairs, which are generally called as root hairs, which is present on epidermis called epiblema, and below this one cortex region is there, that is the outer region, and then endodermis is there. These barrel shaped cells are called as endodermis, and then this is pink color layer is called as pericycle, and this is the xylem and phloem bundles are present. So you can see many xylem bundles are present, many xylem, more than 6, so it is called as polyarch. And in between these xylem bundles, some patches of phloem is present alternatively in between two xylem bundles, right? So it is the clear from the diagram. So all the structures are present here: epidermis, cortex, endodermis, pericycle, vascular bundle and pith, both are all are present, and it has more xylem bundle as compared to dicot root, and usually 6 polyarch condition is there of the xylem bundle. This is the monocotyledonous system, in which the diagram is showing the epidermis. This is epidermis, and below the epidermis there is hypodermis, which was absent in the root, and the function of hypodermis is to provide the mechanical support. These are the vascular bundles, in which you can see this is the phloem and this is xylem. This is phloem, this is xylem. So together they are making the vascular bundle, and these green color all the cells are called as ground tissue. So this is the TS of monocot stem. This is the TS of monocot stem, in which these green color cells show the these green colors structures show the ground tissues which are making the body of the plant, and this is the structure. Now you can see that epidermis is the outermost layer is and single layered uniseriate and covered with thick cuticle, and function of cuticle is to prevent the water loss. Epidermis without trichome. Stems epidermis is without trichome. Hypodermis is present, 2 to 3 layer thick, and it is made up of generally dead cells, that is the sclerenchyma. The function of hypodermis is to just provide the mechanical support, and no differentiation of ground tissue in monocot stem. It means ground tissue is not differentiated into general cortex. You can see that general cortex, pericycle and endodermis, such kind of a structure is not present in them. Cortex, endodermis, pericycle, pith and medullary rays, they are not differentiated. Vascular bundle many scattered in the ground tissue. You can see the vascular bundles scattered in the ground tissue, and generally in monocot stem vascular bundles are many and scattered in the ground tissue. Vascular bundles are oval, that is egg shape, and conjoint, collateral and closed. Conjoint means कि there is no cambium is present in between them. So secondary growth is generally absent in the monocot stem, generally absent in the monocot stem, and phloem lies towards the outside and xylem is inner side. Here in this structure we can say कि phloem is outside and xylem is inside, right? So each vascular bundle is surrounded by scleral chyma sheath called bundle sheath. Yani outside the vascular bundle covering is present. Some of the protoxylem vessels and xylem parenchyma cells dissolve or separate the rapid growth of stem to form a cavity called as protoxylem cavity or lacuna, which stores generally the water. So it is clear from the diagram now. Dorsiventral leaf. Dorsiventral leaf. What do you mean by dorsiventral? Dorsiventral means having two sides, ventral and dorsal sides, that is bifacial. So we have seen in every leaf that there are two surfaces are present which are clearly differentiated from each other, upper lamina and lower lamina, right? So first of all let us understand the diagram. So this is this is the structure, this is the structure in which you can see this is the structure epidermis, this is the epidermis, and this is the adaxial epidermis and this is abaxial epidermis. Two surfaces are there, upper surface is adaxial epidermis and lower surface is abaxial epidermis. This is abaxial epidermis and this is adaxial epidermis, and just below the epidermis there are mesophyll cells are found. So mesophyll cells are of two type, one is elongated called as palisade mesophyll and one is round shape called as spongy mesophyll. So inside the mesophylls you can see these dot structures which are generally the chloroplast containing chlorophyll, and in case of palisade it is present and in case of spongy it is also present. So you can see कि palisade mesophyll is continuous in nature, and in spongy mesophyll there is some air spaces are present, some air spaces are present, and this is the substomatal cavity. Now you can see this is the bundle sheath, this is the bundle sheath, and this is xylem and phloem. This is phloem and this is xylem and phloem. This is xylem and this is phloem, which is making the vascular bundle. Now we can understand this with the help of this text. So bifacial leaf is there, means two surfaces are there and three main parts are there. One is epidermis, this one outermost line, mesophyll, this one, and vascular system, this vascular system. Epidermis is again the outermost single layer of the leaf, covers both the surfaces, which upper surface is called upper epidermis or adaxial and lower surface is called lower epidermis or abaxial. Abaxial epidermis generally bears more. Abaxial, that is the lower one, contains more stomata rather than adaxial. Epidermis is generally covered by layer of cuticle, which is a waxy covering. The function of this is to prevent the water loss. The tissue between upper and lower epidermis is called mesophyll and they are made up of parenchyma having the chloroplast carry out the photosynthesis because they have chlorophyll. In dorsiventral leaf, mesophyll is differentiated into palisade and spongy that I already explained. This elongated cell is palisade and this round shaped structure is called as spongy. So they contain the chloroplast. Now monocotyledonous system. Epidermis is the outermost layer of stem and it is single layered covered by thick cuticle. Monocot stem is without trichomes. Hypodermis is present. Hypodermis is composed of skeleton chyma and provide mechanical support. Now you can see कि these structure are not present, we have already discussed, and bundle sheath is present, that we have also discussed. Bundle sheath is the covering over the vascular bundle. Peripheral vascular bundles generally are present The phloem lies towards the outside and xylem lies towards the inside, that we have seen in the radial vascular bundle, in the conjoint closed and conjoint open vascular bundle. Peripheral vascular bundles generally present are outside are smaller in size, and centrally located vascular bundle are generally larger inside. In monocot stem, the phloem parenchyma is absent and water containing cavities are present, which are called as lacuna. अब these water cavities formed as follows. How they are formed actually? Some of the protoxylem vessels and xylem parenchyma cells dissolve and separate. They dissolve and separate from it. Rapid growth of stem to form cavity called protoxylem cavity or lacuna, which stores water. Now you can see this is the structure of dicot stem. This is the pith, which is the central part of the structure. This is epidermis and this is epidermal hairs. These are epidermal hair which are present on both sides. And this is epidermis, this is hypodermis, which is made up of collenchyma, and this is parenchyma tissues. These are parenchyma tissues, and this is pericycle. This is pericycle. This is pericycle, which is dark green color. Now these are the vascular bundles. These are the vascular bundles, right? So you can see vascular bundle, you can see phloem and xylem, phloem and xylem, phloem, xylem, phloem, xylem. Now you can see कि there is some lines present between these. These lines are actually the medullary rays. These are when they will join, so they will form a ring, and these are medullary rays, which are, and this is the pith or medulla, and this is the outer region. Now the dicot stem, when we see, so this is the epidermis, this is 2 or 3 layers collenchyma cells are present, and then parenchyma cells are present, then the epidermis, and this is the pericycle. This is the phloem, and this is the cambium, and this is metaxylem, which are present towards the cambium, and these are protoxylem, which is present towards the pith. These are metaxylem, these are protoxylem, and this is phloem, and this is cambium. So this will lead the secondary growth, as it is the dicot stem. Now monocot stem, you can clearly see the theory we have done. On outer side smaller vascular bundles are present, and in the center large vascular bundles are present. Here you can see on the periphery there is a smaller vascular bundle, and in the center there is large vascular bundle, and this is green color is showing the ground tissues. This green color is showing the ground tissues, and these are vascular bundles. Here also you can see these diagrams I have already explained. This is vascular bundle, this is vascular bundle, this is ground tissue. Phloem and xylem are present in one structure. They are isobilateral leaves, which are generally found in monocot leaves, and isobilateral leaf, in isobilateral leaf stomata present on both surface of epidermis, hence called as amphioxus, jabki whereas in dicot root stomata is present more in the lower surface of the leaf. Mesophyll is not differentiated into palisade and spongy parenchyma, and generally grasses, inside the grasses adaxial surface is there, and veins modify themselves to form adaxial surface. You can see this is the TS of monocot root, so you can see this is the epidermis, and below the epidermis there is adaxial epidermis. This is adaxial epidermis, and which are generally modified to form the barrel shape structure. barrel shape structure. These cells are called as bulliform cells. These cells are called as bulliform cells. What is the function of these bulliform cells? What is the function of these bulliform cells is just these cells are called bulliform cells or motor cells, and when bulliform cells is present in any leaf, then the main or primary function is just to absorb the water from and make the leaf turgid. Turgid means expanded form. Turgidity is a quality or the feature in which the water absorbed and create the pressure in the cell wall. So the leaf surface exposed, and due to the water stress, if water stress is there means lack of water is there, then bulliform cells become flaccid and make the leaves curl inward so that minimum or less water loss should occur. So in monocot leaf, parallel venation is reflected. Parallel venation means no network is there near the similar size of vascular bundle except in main vein. So this is, you can see the structure. This is the xylem. This is xylem. These all are mesophils which contain the chloroplast. This is the subsidiary stomatal cavity. This is the subsidiary stomatal cavity, and this is the abaxial epidermis. This is the adaxial epidermis in which the bulliform cells are present. Now here the chapter is finished. So kindly do the chapter and do the NCERT back exercise.